Okay, so we are about to dive into a scientific development that honestly sounds like it's straight out of a movie. A breakthrough that could completely change how we think about creating life itself. So let's just get right to the big question. Is it possible to create a human life without using a traditional egg? I mean, for all of human history, the answer has been a very simple, very firm no. But that might be about to change. Get this. Researchers in the United States have done something pretty remarkable. They've made early-stage human embryos, but not in any way you'd expect. They started with the DNA from something as basic as a skin cell.
And let's be really clear about how big of a deal this is. One of the lead researchers, Professor Shukrat Mitailapov, put it bluntly. We achieved something that was thought to be impossible. This is a massive leap forward into territory that until now was pretty much science fiction.
So you're probably wondering, how on earth does this actually work? It's definitely complex, but the idea behind it is kind of brilliantly simple. So let's break down the science of how you go from a simple skin cell all the way to an embryo.
To really get your head around this, it helps to think about something you already know, cloning, like with Dolly the sheep. Now, both processes start in a similar way, by putting a cell's nucleus into a donor egg, but the goal is completely different. With cloning, you're making an exact genetic copy. With this new technique, the goal is to create a totally unique embryo, one that's actually ready to be fertilized.
The secret ingredient here is a brand new process they're calling metamiosis. See, a normal egg has 23 chromosomes, but this new egg, made with the skin cell, has a full set of 46. Metamiosis is the really clever trick they use to convince that egg to just get rid of half of its chromosomes, leaving the 23 it needs to be fertilized by sperm.
So, it all boils down to these 4 steps. First, you transfer the skin cell nucleus into the donor egg. Then, the magic of metamiosis kicks in and halves the chromosomes. After that, the egg gets fertilized with sperm, and finally, a viable embryo, one created without a traditional egg, starts to grow.
This is all really cool science, for sure. But why does it really matter to us? Well, the human impact could be absolutely profound. It could offer new hope for millions of people who are struggling with infertility.
For starters, this could be a total game changer for women whose eggs might not be viable anymore, maybe because of age or disease. This offers a completely new way for them to have a child that's genetically theirs.
And the possibilities don't even stop there. The technique isn't limited to just a woman's skin cells. As we're about to see, you could, in theory, use a man's skin cell to create an egg, which just opens up a whole world of new possibilities for parenthood.
And think about another group this could help. People whose fertility has been damaged by life-saving treatments like chemotherapy. This technology could one day give them back the chance to have a biological family.
But the implications here, they go way beyond just treating infertility. They start to challenge our fundamental definitions of parenthood and what it even means to create a family.
This is where the rulebook of reproduction just gets completely thrown out the window. Because a man's skin cell could be used to create an egg, this could open the door for same-sex male couples to have a child that is genetically related to both of them. Just imagine, one partner's DNA forms the egg, and it's then fertilized by the other partner's sperm.
And hey, don't just take my word for it. This isn't just some wild speculation. Professor Paula Amato, who is actually on the research team, confirmed this is a real possibility, saying that the technology could one day make this a reality.
Now, before we all get way too carried away, we have to remember that this is still really early science. There is a very long, very challenging road ahead, and it's full of big scientific hurdles and some really profound ethical questions that we all need to start talking about.
Let's just start with a quick dose of reality. Right now, the success rate for creating a viable egg this way is only 9%. That is a massive obstacle to overcome before this could ever be used in a clinic.
So why is that success rate so low? Well, there are a few key scientific problems. For one, the egg seems to discard chromosomes randomly, which could lead to genetic diseases. Second, a really important process called crossing over, where DNA gets shuffled around to ensure healthy genes, it's just not happening yet. Fixing these issues could easily take another decade of work.
And you know, these scientific hurdles are a good reminder that this isn't just happening in a lab somewhere. As Professor Roger Sturmy points out, breakthroughs like this demand that we have an open and honest conversation with the public about where this is all going.
And that conversation has to lead to real action. Professor Sturmy also stresses the need for robust governance, basically clear rules and oversight to make sure this is all done accountably. and to build the public trust that is absolutely essential for this kind of research to move forward.
At the end of the day, this technology is a monumental shift, not just in science, but in the very idea of family and society. It really all comes down to one huge question. The rules of parenthood are changing. Are we ready for it?